Hello everyone and welcome to Chapter 13 and Chapter 14 Prep Screencast. Let's start off by going over the breakdown of our peripheral nervous system. So we've already covered the central nervous system in Chapter 12. Chapter 13 will cover most of the peripheral nervous system. We'll talk about our sensory and afferent division, which is going to be information coming into our body through receptors, and motor, or our efferent division. This is information flowing out of our body going toward target organs. And so this area can be broken down into two different parts. We have our somatic nervous system, which this is going to go to our skeletal muscles, whereas our autonomic nervous system, this is going to go to our cardiac muscle, smooth muscle, and also our glands. And we'll talk more about the differences between these two in Chapter 14 when we really dive into the autonomic nervous system. And of course, this system is broken down into our sympathetic division and parasympathetic division. So let's start off talking about some ganglia. There's going to be two major ones that we talk about in our peripheral nervous system. I mentioned one already to you, and that's going to be the dorsal root ganglia. We talked about this within Chapter 12, and that involves part of our spinal cord, which that would be central nervous system, and our dorsal root will lead to our dorsal root ganglia. Now remember that we have only sensory nerves within this dorsal root. And so within our dorsal root ganglia, we're going to find sensory cell bodies in here. Our autonomic ganglia, I'm going to dive into more in our Chapter 14 Prep Screencast and lecture. And these are going to be ganglia that we find along the thoracic vertebral column, and also we have L1 there too. That'll have one as well, but more on this a little bit later. I just want you to know that we find it more in the thoracic area, and it really, let me rephrase this. You're going to find it in a row, in a chain in the thoracic area, although we do have other autonomic ganglia, but they will be located close to or within the organ. So this will make a little bit more sense when we talk about the autonomic nervous system and we discuss the difference between sympathetic and parasympathetic. I just want you to know for now we have two major types of ganglia within our peripheral nervous system. So let's move into regeneration of nerve fibers. So we know that our mature neurons are amitotic. If our soma, the cell body of a nerve, gets damaged, then the axon, though, can regenerate in our peripheral nervous system. And this is coordinated using macrophages to help remove the debris. Those Schwann cells that made those myelin sheaths within our peripheral nervous system, they're going to help regenerate the tube and secrete growth factors to allow the axon to regenerate that damaged part of it. Within our central nervous system, we have those oligodendrocytes that are going to bear growth inhibiting proteins that prevents central nervous system fiber regeneration. But of course, as I mentioned in class, this might be changing as far as what our research is telling us. So be on the lookout for some new information coming out regarding that. So let's look at these steps here. Here's an area where we have nerve damage and our axon is fragmented. We can see we have droplets of myelin here. The axon is just completely destroyed here. So our macrophages come in, they clean out the debris. Then we're going to have our axon sprouting these nice fine filaments all over the place, and eventually those are going to start to not fuse together but come together, and our Schwann cells are also helping form that myelin sheath around it. And so here is our nice new axon with the myelin sheath formed around it. Now I want to dive into our cranial nerves. We have 12 pairs of them, and I mentioned in lab that there's quite a few sayings out there, mnemonics, that'll help you memorize these. This one that I have right here will help you memorize each of the cranial nerves and whether it's sensory, motor function, or both. And the saying is, some say marry money, but my brother says big brains matter more. So for example, cranial nerve number 1 would be S for sensory. Cranial nerve number 3 would be M for motor, although it technically does both, but it's more motor function. But anyway, another way you can remember the names of the cranial nerves is to learn phrases like this. On occasion, our trusty track acts funny, very good vehicle anyhow, or oh, 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 to touch and feel, very good velvet, ah, heaven. So each letter in the beginning of these mnemonics stands for the name of our the name of our cranial nerves. They might work for you. There's also a ton more out there on the web. I think Wikipedia has a page that lists like 50 different types of mnemonics to learn the cranial nerve. So use whatever you want. In class, I'll be telling you some stories and ways that I remember my cranial nerves, or how I learned it in the past. Now I've just committed it to memory, but it might help you, it might not. It'll help you to apply it to your life in some way. Okay, so before I show you, I think, hold on, let me see this next slide. Oh, okay. Here's the slide I want. I don't know how I ran past this. So I'm going to go through step by step and talk about each cranial nerve and just a little quick summary about it. So you can create a chart for yourself, or you can put it, I think it's page 33 in your RMU lab manual, just to jot down a quick summary of its function, and then we'll go into more detail in class. So first we have cranial nerve number 1, and we see the olfactory bulb here. The nerves are going to come off of the bulb and enter into the nasal cavity through the foramina. Foramina means multiple holes of the cribriform plate in the ethmoid bone. And this nerve is in charge of the sense of smell. Cranial nerve number 2, which is our optic nerve, this is coming off of the eyeballs. So our eyes would be right here, let's say, and then so the optic nerve would move on back this way, and eventually what they're going to do is cross at a place called the optic chiasm here. After they cross, then it becomes the optic tract, and it's going to go back and synapse in the thalamus. There's a specific nucleus there in the thalamus that it synapses with. And remember, the thalamus is our sensory relay center. So any type of sensation that we're getting has to make a pit stop there, except for our sense of smell that has a different pathway. And after it synapses at the thalamus, then it's going to travel through optic radiations over to our occipital lobe, which we can't see here because the cerebellum is covering it. Okay, moving past cranial nerve number 2, we have cranial nerve number 3, which is oculomotor, and this is going to supply 4 of our 6 extraocular muscles. Extraocular muscles are going to help us move our eyeballs around, so looking to your right, to your left, up, down, it's assisting with those movements. So I'm going to show you real quick. This is an example of intrinsic eye movement, where the iris is helping control how much light moves into our eye through the pupil. So it can constrict to allow less light in, let's say in bright daylight, or at night or in dim lighting, we would have a dilated pupil to allow more light in and help us see in darkened rooms. And this is an example of our extrinsic eye movement. Eyeballs just moving all around. But of course, we want our eyeballs to function together. So let's move into our trochlear nerve. This is the smallest of all the cranial nerves. This is cranial nerve number 4. And this is in charge of supplying just one extraocular muscle. And that muscle is the superior oblique. By the way, we're going to talk about the names of each of these eye muscles in our sensus lecture, chapter 15. Now, cranial nerve number 5 is the trigeminal nerve, and this is a really cool nerve. It's the largest cranial nerve, and it divides into 3 different parts. So I mentioned to you in lab, well, most of you in lab, if not all of you, that you can take your 3 fingers and hold it up to your face toward your eye, maxilla, and mandible, because we split them into V1, V2, and V3. So V1 would be ophthalmic, pointing to our eye, V2 would be maxillary, pointing to the maxilla, and V3 would be pointing to our mandible. And so these 3 nerves are going to give sensory supply to the skin there too, in those regions. And we're also going to give innervation to muscles of mastication. And it's going to be V3, the mandibular nerve, that specifically gives that innervation to the muscles of mastication. Cranial nerve number 6, abducens, right over here. That's only going to supply one eye muscle as well, and that eye muscle's name is lateral rectus. And then we have cranial nerve number 7. Where's 7 for me? Over here. Before I move into talking about cranial nerve number 7, we're going to talk about this in lecture, but cranial nerves number 3, 4, V1 of 5, and cranial nerve number 6, all 4 of those are going to exit through an area called the superior oblique fissure, which is going to go into our orbit area. Our facial nerve, cranial nerve number 7, as well as vestibulocochlear, cranial nerve number 8, both of those are going to exit through the internal acoustic meatus. And what I mean by exit is that all of these are really coming off of the brainstem, except for cranial nerve number 1 and 2. So they're coming off of the brainstem, and then they have to exit through, and I'm going to go ahead here real quick. They have to exit through an opening of our skull in order to travel to areas of our face or our neck, chest, or abdomen. So that's what I mean by a cranial exit. So for example here, we have the optic nerve, and this is exiting at the optic canal. So underneath this bone here is our eye, and this optic nerve is coming from the eye going into our cranial cavity because it needs to convey that information to our brain. Whereas some of our other nerves, we have more fibers going on out. So like our, let's say V3, our mandibular nerve of our cranial nerve number 5 that supplies muscles of mastication, that information is flowing out. And so we need to go through a specific exit to move down toward those muscles in our face. All right, let me go back here. Okay, so what does our facial nerve do? This is going to supply our muscles of facial expression. So if you're frowning, smiling, raising your eyebrows, facial nerve supplies those muscles. It'll also supply our lacrimal glands and salivary glands, and also part of our tongue as far as taste. Vestibulocochlear, that is going to deal with hearing and balance. So vestibule is referring to the balance portion, cochlear is referring to the hearing portion. Now cranial nerves number 9, 10, and 11, these 3 are exiting through the jugular foramen, and I'll show you where that is in the next slide. But first let me tell you the functions. So glossopharyngeal, that is going to give innervation to the pharynx to help us with swallowing. Also, it gives innervation to part of our tongue and gives parasympathetic innervation to our parotid salivary gland. Next we have vagus, and vagus is the longest of all of our cranial nerves. This one travels from this medulla all the way down through our thorax and through our abdomen, because it's going to have parasympathetic innervation to our thoracic and abdominal organs. So for example, our heart or the stomach. Next we have the accessory nerve, and this is cranial nerve number 11. This too is coming off of the medulla, but we also have nerve fibers down here that are originating from our cervical portion of our spinal cord. It's actually from the ventral rootlets of C1 to C5. And so what it does is it originates there, and then it re-enters through the foramen magnum back up here, and then exits out the jugular foramen to go and supply, and this is its function, the sternocleidomastoid muscle as well as the trapezius. And lastly, we have our hypoglossal nerve, which also comes off of the medulla, and it's going to exit through the hypoglossal canal. And its function is to give motor supply to our intrinsic and extrinsic tongue muscles. So it's going to help us a little bit with swallowing with the pharynx as well. So I want to show you this image here because this is what you guys are seeing in the cadaver lab. It's just illustrated through one of my favorite anatomy illustrators, Frank Netter. And when you're looking at the skull, this is the anterior portion, this is the posterior portion. Yes, we have some portions of the dura mater in place here, and we can see those venous sinuses as well. So all of these in blue, these are venous sinuses. And I want to move anterior to posterior to kind of explain what you're going to see in the lab. So you shouldn't really see the olfactory bulb here because it stays on the brain. So let's start here at cranial nerve number 2, the optic nerve. In lab, I mentioned to most of you that I like to picture a little box here. There's my box. And this looks a little bit different because they've peeled away some of the fascia here. So at the top of your box on the outer corners, you're going to find first the optic nerve. And then right behind that, you're going to find the internal carotid artery. If you look at the center of the box, you're going to find the pituitary gland, or a little bit of the stalk of the pituitary gland. And then in this little edge here, I should have made my box a little bit longer. Okay, more like that. At the bottom edge, you're going to find cranial nerve number 3, which is the oculomotor nerve. Now in the lab, we're not going to see the trochlear nerve. That is going to be our smallest cranial nerve. Usually gets pulled off when we pull the brain out, but for those of you that stayed until the end of lab, you were able to watch me take the one of the brains out of the cranium, and we actually had a trochlear nerve there. But then in my attempt to open up the tentorium cerebelli a little bit more and cut some of that tissue away, I accidentally cut off the trochlear nerve after I had found it and showed it to you guys. So anyway, you won't see that in lab. It won't be on your practical. Next, we have trigeminal, and what you guys will see in lab is this area will be more covered, so you won't see this area that has the 3 divisions. Instead, you'll just see this one big thick bundle here. And so the next thing what you're looking for will be more of this, I call it the petrous portion of the temporal bone. Petrous means rocky, so this is a rocky area in here, and I know this is starting to look messy, so I'm going to clear this off if this lets me. Okay, so anyway, we're just looking from here on back. So look at that rocky portion there, and then look for this thick nerve. That's going to be number 1. So I'm going to put a 1 there. The next thing you're going to see is an opening here beyond this ridge, and that's going to be number 2. Number 2 is going to be your internal acoustic meatus, where we'll have cranial nerves 7 and 8 exiting, and then below that region, that's your 3rd opening beyond your petrous portion, and that is going to be the jugular foramen that contains cranial nerves 9, 10, and 11. Now, you might say, well, is there going to be a 4 now? Yes, but I don't really count this as 4 because it's easy to find. You're going to see the foramen magnum here. I should make that a little bit smaller. Right above the foramen magnum, you're going to have this opening on the lateral side, and that's going to be your hypoglossal canal that contains the hypoglossal nerve. So there you go. You have cranial nerve number 2, optic nerve, cranial nerve number 3, oculomotor. We're going to skip 4. 5 is that big thick one. I forgot to mention cranial nerve number 6. This you're going to find more in the midline of the cranium. So this is cranial nerve number 6, abducens here. And I'm gonna cross this part off because you're not gonna see this long portion. You're just gonna see it coming off more towards the middle here, sticking out. And then from there, you locate your petrous portion and find your trigeminal nerve, 7 and 8, 9, 10, and 11, and lastly, hypoglossal. Okay, that brings me to your spinal nerves. So we have 31 pairs of our spinal nerves, and the number of these spinal nerves will correlate with the number of vertebrae, except for, cranial nerve, let me start over again. You'll have the spinal nerves correlating with the number of vertebrae, except for your cervical spinal nerves, because we have 7 cervical vertebrae, but we have 8 cervical spinal nerves. And that's because we have one extra one that comes off of the top. So we're going to have the rest of these correlating. 12 thoracic vertebrae, 12 thoracic spinal nerves, 5 lumbar, 5 sacral, and 1 coccygeal. So what I want to do is talk about how these nerves are moving in and out. So I'm going to grab my pen real quick. Okay, so we want to label these areas and talk about exactly what they do. So this is kind of what we did in class. I just want to kind of review. So we have our gray matter here, and our gray matter, that is going to contain cell bodies. And we also have some unmyelinated fibers in there. Let's say unmyelinated axons. Our white matter, this is going to contain myelinated axons, but we also have some unmyelinated axons in there as well. It's just we have more of the myelinated ones, and that's why this appears so white to us. Now next we have our dorsal root. The dorsal root contains only sensory nerves. Okay. And then this, and of course these colors are corresponding with the names. This is going to be our dorsal root ganglion. This contains only sensory cell bodies. Okay. We have next our mixed spinal nerve, and this is going to contain both. I'm going to put M for motor and sensory nerves. We have both in there. So sometimes instead of it just being called, do I not have a label for this? I guess I don't. Let me write this in. Sometimes it's called mixed spinal nerve because it has both motor and sensory nerves in it. Other times we just call it a spinal nerve. So this is what we're talking about when we said we have like 8 cervical spinal nerves. So this could be C2, for example, cervical spinal nerve 2. And then the next thing I'll show you is going to be our dorsal and ventral ramus. Our dorsal ramus is actually smaller, and that's because this only supplies back structures. Okay. And then we have our ventral ramus, and this supplies our trunk. So really it's the anterior trunk and limbs. Okay, so that kind of tells you what each part of our spinal cord is supplying. Now I want to talk about our nerves that are going to move through and how they move through here. So let's do blue for motor. This is going to be my motor neuron. And let's see, I think we could do red here for our sensory neuron. Okay, neuron. So we always say that sensory information is coming in. So what I want to do is talk about an example that I mentioned in class, and then we'll do one more. So you put your hand on a hot stove, right? So first we have to have information moving into our spinal cord. So way out here is, let's say, our hand, and we're going to have specific nerves like, let's say, our median nerve, which we're going to learn about later. That information needs to flow through the median nerve, and it's going to go into our ventral ramus because it's coming from a limb, okay? Because this sensory nerve, it could come in through our dorsal ramus or the ventral ramus. But because it's not our back that's being burned off from the stove, that's why we're not using this one. So we're going to continue through our ventral ramus, and then that's going to go through the mixed spinal nerve. And then we have a choice of going through either the dorsal root or the ventral root. Because only sensory nerves go through the dorsal root, we're going to choose that door. And then we're going to stop here in the gray matter. And then this is going to be a unipolar neuron, and that's why we have a cell body that sprouts out from the axon here, and we'll just have a collection of these cell bodies here at the dorsal root ganglion. They're not synapsing here, they're just moving on past. Now, this is going to be kind of more of a reflex. So what was my color for motor? Blue. So it's going to synapse onto my motor neuron that starts in here in the gray matter, and then we have to go out the ventral root because this contains, oh, and I forgot to write this in, this contains only motor nerves. Okay, so we have to go through that door. So let's continue on now, and this has to go through our mixed spinal nerve, and then we have a choice to go out through our dorsal ramus or ventral ramus. Our choice is going to be ventral ramus because we want to supply the muscles of that hand or arm to pull it away from that hot stove. So we leave out that way and go to those muscles. Okay, so let's do one more example, and I don't know that it's going to let me erase only my nerves. Let's see here. Sorry, I don't want to waste time in our screencast. Erase pen. Perfect. Oh, okay. So it just got rid of everything. Anyway, here's motor. Let's go back. Here's sensory. So now if we get stabbed in the back and then we use our biceps muscle to flex our arm and whatever, we're using our limb muscles to reach on back and pull that knife out of our back, let's say. A little aggressive here, but it's fine. So our sensory information, that's going to come in through the dorsal ramus this time because we got stabbed in the back, and that supplies back structures. We go through the mixed spinal nerve, and then we have to go through the dorsal root because only sensory neurons are going to move through there. Okay, and then let's not forget our little cell body, and then we're going to synapse with our motor nerve, and that has to exit through the ventral root out because we only contain motor neurons here, out the spinal nerve, and then through the ventral ramus out because we have to synapse with our limb muscles in order to pull that knife out. So those are just some examples. We could do more in tutoring if you'd like. Okay, now a bunch of our, actually before I move on, a bunch of our ventral rami, they are going to create plexi. So they're going to come together and intertwine all of their nerves to create plexi. And of course, with anything in anatomy, there's always an exception. So our exception is T, oh, why is my pen not working? T2 through T12 are not going to create plexi. Instead, they are going to create intercostal nerves. intercostal nerves, and they'll go to supply muscles of the ribs, the anterolateral thorax, and also the abdominal wall. And then the last thing I want to mention on this slide is that T1 through L2 are going to have rami communicantes. So I'll talk about this more in our autonomic nervous system lecture, but our spinal nerve, this mixed spinal nerve here, is going to give branches to the dorsal ramus, the ventral ramus, and then we also have something called the white ramus and also a gray ramus. And we're also going to have this meningeal branch that's going to go back in to our vertebral column to give supply to the meninges here. But anyway, these rami communicantes, they're going to work within our autonomic nervous system. So I'm not going to talk about them now, but later on in this prep screencast. So let's talk really quickly about these different plexi. We're going to talk about 4 of them total. Your first one is going to be your cervical plexus. They come from the ventral rami of C1 to C4, and they're just going to give branches to a bunch of different nerves that'll supply the skin and muscles of the area of the neck, the ear, the back of the head, and the shoulders. But the main, most important nerve I want you to know about is the phrenic nerve. And of course, you guys are familiar with the phrenic nerve already. That is going to give major motor and sensory supply to our diaphragm. And remember that saying, C3, 4, and 5 keeps the diaphragm alive. That's the ventral rami that this nerve comes from. So you could see this coming off here and then descending on down across the side of the heart, the mediastinum really, and down to the diaphragm. And irritation of this will cause hiccups or spasms of the diaphragm. Now we have our brachial plexus, and you really want to thank me for not having you memorize all 16 nerves that come off of the brachial plexus. But I do want you to know that this is going to come from the ventral rami of C5 through T1. So I can't see here. Oh, there's C5 for us, and all the way down to T1. And you could see this is branching extensively here. So we break this down into different segments. So this first segment is going to be the roots, and this comes from the ventral rami of C5 to T1, like we said. The trunks are going to be the upper, middle, and lower, and then they divide out into anterior and posterior divisions. So posterior divisions here are pictured in the green, whereas anterior is pictured in this yellowish color. They just don't show it to you. Anyway, from there, we are going to finally get our chords, and I want you to know your chords for your exam, and that's going to be made up of a lateral, medial, and posterior. So here's lateral, here's posterior, and here's medial. Basically how you would describe their orientation to the body, because here's our midline, this would be medial, and this does sit more posteriorly. We're not going to be looking at this in the cadaver lab. We're going to see this portion more in the armpit region. And then I'm going to show you how to find your M in the armpit, and we're going to learn about these major nerves that come off. So let's just break this down a little bit. Here's your roots. There are your trunks. Here's your posterior divisions. Here are your anterior divisions. So you can see how they kind of branch. Like here are your anterior divisions, and if we trace this back, we can see, oh, here's C5, C6, C7, C8, and T1. So when we learn your chords, like lateral, posterior, medial, your lateral chord, any nerve that comes off of there, so let's say your musculocutaneous nerve that comes off of there, you could trace this back and say, oh, look, it's going back this way and this way. So this musculocutaneous nerve comes from the ventral rami of C5, C6, and C7, whereas something down here like your ulnar nerve that we're going to learn about in class, if you trace that back, we only really come from C8 and T1. So these would be your terminal branches. So if you wanted to kind of practice and knowing how to break down your brachial plexus. You can write out your ventral rami, and I like to start from the chords because that's all you need to know. So there's your lateral chord. That comes from C5 to C7. Your posterior chord comes from all of C5 to T1, and your medial chord comes from C8 to T1. Then your posterior chord is going to give rise to one of the nerves I want you to know for your practical exam, your radial nerve. And your lateral chord and medial chord are going to create roots. So they come together, and we call this first branch over here your lateral root of the median nerve, and this one is your medial root of your median nerve. And then that, of course, creates the actual median nerve. And then we have this branch, the musculocutaneous nerve, coming from your lateral cord. And lastly, your ulnar nerve coming from the medial cord. So what we're going to do in lab is we're going to look for this M, and here it is. This is the M that we're finding in lab. Your lumbar plexus is going to come from L1 to L4, and it's going to innervate the thigh, abdominal wall, and psoas muscle. And so I want to show you your two major nerves. You have your femoral nerve that comes from L2 to L4, and that innervates really all the muscles of your anterior thigh and also the skin of your anterior thigh and medial surface of the leg. And then your obturator nerve is the other big one, and that supplies all of the muscles of your medial thigh. And these are some other large branches that come off of your lumbar plexus. You have your iliohypogastric nerve, and this is going to give a sensory supply to the skin over the lateral gluteal region and also right above the pubis. And its motor function is to supply the internal oblique muscle and transverse abdominis within the abdomen. Your ilioinguinal nerve, you can see here coming from L1 as well, and also down over here. This is going to supply in a male, the skin over the root of the penis and part of the scrotum, and in a female that's going to supply the skin on the mons pubis and the labia majora. So I want you to know those for your exam. And then lastly, we have your sacral plexus, and this comes from L4 to S4, and this will supply basically all of our lower limb, as well as our pelvic and buttock structures and the perineum. The major nerve I want you to know here is the sciatic nerve, and this is going to be the longest and thickest nerve of the body. It'll innervate the hamstring muscles, part of adductor magnus, that extensor part, if you remember that from our muscular lecture, and also most of the muscles of the leg and foot. But really, this sciatic nerve, the tibial part, is going to continue to supply the posterior leg, whereas the common fibular is going to supply the lateral and anterior leg. Okay, so this tibial and common fibular nerve is surrounded by a common epineurium. So remember, that's the outer layer of a nerve. And it binds it together. So when we're in lab, if you really wanted, you can take your sciatic nerve and divide out these two portions of the sciatic nerve. So I have this diagram here, but I always like it. I always feel like it's easier to talk about when you draw it out yourself. So moving on down from your sacral plexus, you have your sciatic nerve. And then what your sciatic nerve does at the level of the knee, you can see it here, it splits into a common fibular nerve and also your tibial nerve. So let me change colors here to make this a little bit easier to see. Your tibial nerve, after it passes the knee, is going to go down and supply all the posterior leg. So this is my tibial nerve. Whoops, tibial nerve. And then let's go back up and talk about that common fibular nerve. I wish I had more space here. I'm going to abbreviate common fibular. And remember that the older term for fibular is peroneal, so don't be confused if you see that anywhere. It's the same exact thing. So this common fibular nerve, what it does is it splits into Great. Let me redraw this. Okay, so I redrew this here. So sciatic is coming down through the posterior thigh, and then at the back of the knee, it's going to split into your tibial to continue down the back of the leg to supply all those structures. And then the common fibula, what it does is it wraps around the head of the fibula and it starts to branch into two other nerves. It branches into the superficial fibular, which will go and supply the lateral compartment of the leg, and then the deep fibular will move over anteriorly to supply the anterior leg structures. And then our tibial, I didn't draw this out, but our tibial nerve is also going to give branch to our plantar branches. You won't have to identify these in lab, but there's a medial and lateral plantar nerves that are going to give supply to our foot. Now we're going to talk about our reflexes, and we have really two different categories. We have inborn or intrinsic reflexes, like the one you're seeing here in our eye of our iris, where it's involuntary and it's a predictable type of motor response, whereas our learned or acquired reflexes, that takes practice and repetition. So put a picture here of Paul Walker, known for those Fast and Furious movies, because he has practiced through repetition and improved his driving skills. So let's talk about this reflex arc. We talked about it when we discussed putting our hand on a hot stove. So let's break it down into 5 different components of our reflex arc. So we have our receptor, which in the example we used, it was a thermoreceptor within our hand, but in this illustration, we're going to use a pin here. So let's say it's a nociceptor or a pain receptor that's picking up this information, and it sends it through, number 2, our sensory neuron, which is sending afferent or sensory impulses to our central nervous system. Then we have our integration center, which this can be either monosynaptic or polysynaptic. Mono meaning one synapse means that this sensory nerve is directly going to synapse onto this motor nerve. That's not the case in this image. This image is showing us polysynaptic, meaning we have multiple or many synapses. So this sensory nerve is synapsing with the interneuron first, and that interneuron is then carrying it over to the motor neuron, which is number 4, and that is sending the efferent or motor impulses from our integration center over to our effector organ, which in this case is a skeletal muscle. So our skeletal muscle would be number 5, the effector organ that'll respond to that motor impulse by contracting, or if it was a gland, it would secrete something. Our spinal reflexes are going to involve our integration center as only our spinal cord. So instead of going up to the cerebrum and back down through the spinal cord, everything just takes place in our spinal ord. And our effectors are going to be our skeletal muscles. And we can test these somatic reflexes within a clinical setting to assess how the nervous system is functioning. So let's talk about our stretch reflexes first. This is when you go to the doctor and they tap your patellar tendon to see if you get the reflex of the knee jerking up. So let's talk through this step by step. The first thing that happens is we have muscle spindles within our muscle, and our muscle spindles are basically telling our brain the length of the muscle. So we start to get a sense of stretch there. We get type 2A sensory neurons that are going to send this information into our spinal cord, and they're going to synapse directly with the alpha motor neuron, and so this would be a monosynaptic reflex. And so we're moving through this motor neuron, and that goes over to our quad muscle, causing it to contract. Now when it contracts, that is going to cause our leg to extend. So it moves it back on up, so our limb would be straight here. And this is always going to be ipsilateral as well, meaning this reflex will always be on the same side. The other thing that's going to happen is called reciprocal inhibition. So we're going to get these same type 2A sensory fibers that are going to move into our spinal cord, but instead of this time synapsing on these alpha motor neurons, at the same time they're synapsing with an interneuron. So now this is a polysynaptic type of reflex here, and then it'll synapse with this alpha motor neuron, but this alpha motor neuron is going to our hamstrings. And so this is causing these hamstrings to elongate and allow our limb to move on up. Basically, it's saying you're not going to contract, you're going to relax and allow the leg to extend. Our Golgi tendon reflex is going to be similar but a little bit different. Our Golgi tendon organ is telling our brain the amount of tension in our muscle and in our tendon. So our quad is going to contract, that activates our GTO, or Golgi tendon organ. And so that information is moving along a sensory neuron into our spinal cord and synapses with interneurons. So this is always going to be a polysynaptic type of reflex. Then we move through an alpha motor neuron to our quadricep muscle, and that is going to cause the muscle to relax, whereas the other alpha motor neuron going to our hamstring is going to cause the muscle to contract. We also have flexor and crossed-extensor reflexes. This is when something, usually that's painful, is going to cause our limb to withdraw or move away. And this is always going to be ipsilateral and polysynaptic. This is our crossed-extensor reflex, where if we have a painful stimuli, let's say to our right lower limb, that's going to cause the flexor reflex. So we're going to flex our limb to pull it away like you're seeing here. But it also causes a contralateral reflex in which we are going to extend this left limb here. I hope I'm using the right side here, where we're going to extend that in order to be able to pull that flexed limb away. We have superficial reflexes as well, and all of our superficial reflexes are going to be some type of gentle cutaneous stimulation, and this reflex is going to depend on our upper motor pathways and the cord level reflex arcs. So this is something that I actually tested on my son myself, of course, because what you do is start down here at the heel, and not like a sharp object, but maybe like a pen cap or something, and draw it on the lateral side of the foot up toward the toes. And this should cause, in a baby, I'm going to switch screens here. In a baby, it should cause a Babinski sign. So we're going to have the toe moving on up and away, as well as the toes kind of fanning out. But in an adult, there we go. In an adult, we should have the toes kind of curling forward. And this is showing us, I should say, the toes should be flexing downward. That's what I meant. And this is showing us whether these corticospinal tracts within our brainstem and our spinal cord are all myelinated and functioning correctly. What a corticospinal tract is a group of fibers that are moving throughout the white matter here over to make a connection to our cerebrum. So this is in charge of our discrete voluntary skilled movements like our fingers and toes. And this information is all motor. So it starts out in the cortex, and then it's going to move through our midbrain, eventually through the pons and our medulla, through our spinal cord at whatever level it needs to function. So eventually, I believe it's after the first year these tract fibers should all be myelinated. So then you can test this out on a baby and stroke the lateral side of their foot, and they shouldn't be exhibiting this Babinski reflex anymore. They should be curling their toes downward. Okay, so now we're going to go into chapter 14, and so I'm going to try and keep this a little short. We'll talk about details in class. But so our autonomic nervous system is broken into a sympathetic and parasympathetic division. So let's talk about some of the differences between this. Of course, your parasympathetic nervous system, you know this as your rest and digest response, whereas your sympathetic, that is your fight or flight response. And within our autonomic nervous system, we're always going to have two motor neurons or nerves. Whereas, let me go back one slide, it's going to get rid of some of my text, but whereas with our somatic nervous system, this is always just going to involve one motor nerve. So what this means is, here's our spinal cord, there's my H, okay? We have one motor nerve that's going to leave through my ventral root, through the spinal nerve, and out whatever rami, and go to supply my skeletal muscle. No problem, nice and easy, one nerve. With our autonomic nervous system, what you're seeing here is we are going to have, let's go with this one here, we're going to have our first motor neuron called the preganglionic neuron leave the spinal cord, and then it's going to synapse with an autonomic ganglia. Sometimes this is called sympathetic ganglia within our sympathetic nervous system. And from there, our second motor neuron, or this is also known as our postganglionic neuron, is going to leave that and travel over to its target organ, in this case the lungs. So one of the main differences between our parasympathetic and sympathetic is that our sympathetic ganglia or autonomic ganglia, it's going to be located close to the spinal cord. Okay, let's put ganglia. Okay, these ganglia are all located close to the spinal cord. In fact, they're all kind of lined up in a row here. So we like to call this the pearl necklace in the cadaver lab, because you just have a bunch of these little strands of balls moving down the vertebral column. So I'll show you that in lab. That's going to be on your practical exam. The other thing is that this is a part of the thoracolumbar system, meaning that we only get nerve fibers from the thoracic and lumbar region here. What else did we need to mention? We'll talk about some of the functions in a moment here too. Now let's talk about our parasympathetic nervous system. That's what I wanted to mention. So if you look at these preganglionic fibers, I'm just going to abbreviate this as pre-G. Preganglionic fibers in our sympathetic nervous system are going to be short, whereas postganglionic fibers, they're going to be long. So if we look at our parasympathetic nervous system, this is going to be the opposite for all of these categories. So if we look at our ganglia, these are going to be far from the spinal cord. In fact, where we're going to find them are close to or within the organs themselves. So look right over here. This is an autonomic ganglion. This is an autonomic ganglion. This is an autonomic ganglion. This one here, here. They're all mostly located inside the organs. And the other thing is, instead of the thoracolumbar system, this is coming off of cranial nerves or sacral. So we call this the craniosacral system. And then we're going to have our preganglionic fibers as long and our postganglionic fibers as short. Look how short they are. It goes from here to here because they might be located within the organ themselves. Okay, so those are the main differences as far as composition. Let's talk about some of their roles within our autonomic nervous system. So here's our parasympathetic division. This is the rest and digest response. So they are basically conserving our body energy, and they maintain everything that's going on when we're at rest. So this is like us digesting our food, secreting gastric juices, salivary glands are functioning, giving more saliva. Peristalsis is working in segmentation within our intestines, and we also have increased glucose uptake from our blood. So our pancreas is assisting us in that. And then our pupils are also going to remain constricted, and our lenses are going to accommodate for close vision here. And it involves our ciliary ganglion. We'll talk about this ganglion in a moment. Our sympathetic division or fight or flight system or response, this is going to deal with when we have that adrenaline rush, when we're threatened, when we're exercising. And so we're going to need our blood flow to move over to our skeletal muscles and hearts to help us with either exercising, running away, or fighting. Our bronchioles are going to dilate, so we'll have an increase in respiratory rate, and our liver is going to release glucose in order for our body to function optimally. Okay, now here's some of those ganglia that I mentioned, like the ciliary ganglia and our parasympathetic nervous system. These are kind of the outflows of how certain organs are controlled. So I'm just going to talk about one of them here. We'll talk about the rest in class. So here's oculomotor number 3, and we could see that coming off of the top of the medulla, of the midbrain. And this is our preganglionic nerve coursing on down. We have a synapse at our ciliary ganglion, and then that postganglionic neuron is going to go over to supply the eye. And this will help with the pupil constriction that we were mentioning earlier. So anyway, we'll talk about the rest of these in class. Now I was going to talk about the different pathways in which our sympathetic trunks are going to course, but I'm going to talk about 2 and 3 in class. What I want to just tell you about is those 2 rami communicantes that I mentioned earlier in our peripheral nervous system. So here is our spinal nerve, and we could see our ventral ramus coming off here, and this is going to be our white ramus, and this is our gray ramus. So our preganglionic neuron, let's just follow it. It leaves the spinal cord, goes through our ventral root because it's a motor nerve, right? Through the spinal cord, through the spinal nerve, and then it's going to exit out the white ramus and synapse at an autonomic ganglion. This autonomic ganglion, let me take you back, is what we're seeing right here. This is where that synapse is happening. It's just this is a little bit more of a closer, more detailed view. If I can get to that slide again. Okay, so that's what's going on here. We're synapsing in here, and then our postganglionic neuron is moving out through the gray ramus and leaving out the ventral ramus to go over to our effector organs. So like I said, I'm going to talk about these other pathways later on. This is just another illustration of that happening. We can see the preganglionic neuron exiting here through the white ramus, and then here's our sympathetic ganglion where it's going to synapse, and then the postganglionic nerve fiber is leaving through the gray ramus, and then that'll leave back out through the ventral ramus. We're going to talk about these collateral ganglia and splanchnic nerves in class. Okay, so sympathetic tone. What's interesting is our sympathetic nervous system controls blood pressure at all times. So even though we're at rest, your sympathetic nervous system can still be functioning. It's just not functioning to the extreme of an adrenaline rush. So the way it's going to do this, and let me switch to this next slide here, is that when we need to create more high blood pressure, we're going to have our sympathetic fibers firing more rapidly in order to increase our blood pressure. Whereas when we're at rest, we don't need our we're relaxed, we don't need our blood pressure high, then we have these sympathetic fibers fire less rapidly. And we can also have alpha blocker drugs interfere with these vasomotor fibers to treat hypertension. And here are some cooperative effects. So this is best seen in control of our external genitalia. So we have parasympathetic fibers causing vasodilation that are going to be responsible for the erection in a male taking place to allow all that blood flow into the penis and also in the clitoris of a female. And that'll also allow for vaginal lubrication. Sympathetic fibers are going to cause the ejaculation of the semen in a male and the contraction of the female's vagina. So that's going to be it for your prep screencast. I know it was like an hour, but we did 2 chapters within this hour.